Okay, on to part two. So last time we talked a little bit about meiosis, and now we're going to move on to genetics. And the reason that those two things are coupled in this lab is because meiosis and genetics are related phenomena. So we talked about a fungus named Soldaria last time. I want to switch gears right now and talk a little bit about pea plants. Okay, so hopefully you remember from high school biology that there was a dude named Gregor Mendel. We consider him to be the father of genetics. And he figured out a lot of what was happening through rearing pea plants. And, excuse me, one of the traits that he examined in pea plants was seed shape. And so for my next little few explanations, let's think about seed shape as having two forms, okay? You can either be a round pea, which we're going to designate as capital letter R, or you can be a wrinkled pea. Sorry, I should say wrinkled, a wrinkled pea, which we are going to designate as little r. Let's look at how meiosis explains a lot of genetics. So I'm going to draw a cell like I did last time, and I'm going to place in it two chromosomes like I did last time. Okay, and on these two chromosomes, we are going to have the gene for seed color, and we're going to have big R on this chromosome and little r on this chromosome. So we have one gene for seed colors, but we have two different forms of the gene, and different forms of the same gene, I'm hoping that you remember, are called alleles. Okay, so we have two different alleles. Now, if we undergo meiosis 1, we're cutting it down the middle. So that's our meiosis 1, our reductional division. That's going to take us from 2n, which is a diploid scenario, because here we have one cell with two copies of the same chromosome. Meiosis 1 is going to take us to a haploid situation. We're going to cut that in half. We're going to pull this one over. We're going to pull that one over. We're going to end up with two cells, right? This one is going to have a single chromosome in it. We know that because there's one centromere with the big R allele, which is for round C shape. This one is going to have a single chromosome in it with a little r allele for wrinkled C shape. And if we undergo meiosis 2, so this is going to be 1, this will be meiosis 2, you can see that we'll end up with 4 cells eventually. Right? Each of them having a single chromosome. This one and this one are the result of this pulling apart. So they're each going to have the big R allele. And this one and this one are the result of that pulling apart. And they're each going to have the little r allele. Okay? So we've talked, you've probably seen a lot of like big R, little r stuff, and where that comes from is all from meiosis, okay? This mama here has two options for what she can give to her offspring. She can either give them this allele or that allele, and that is all because of meiotic cell division. Now, let's talk about Gregor Mendel for just a second. One of the things that he did was he figured out some principles that are going to help us figure out our problems later. So he had a whole bunch of pea plants that he bred time and time again, generation after generation. And he found that these pea plants were always round. And then he had a second variety of pea plants that he bred generation after generation, just time and time again, and they were always wrinkled. Okay, the peas were always kind of shriveledy looking. So Gregor, our buddy, back in the 1850s, asked himself what would happen if he actually crossed these two pea plants. what would their offspring look like? He actually predicted that there would be some sort of intermediate mingling, like maybe you'd have a slightly round, slightly wrinkled, blended kind of seed. But what he found instead was that all of the offspring from this were round. So that leads us to the very first thing that Gregor discovered, and that is the principle of dominance, or we'll just say dominance, okay? The principle of dominance. So you remember the word dominant and recessive in relation to genetics, right? So in this case, round covered up the wrinkle. There was no trace of this wrinkled trait. So round was dominant to wrinkled, and we'll say that wrinkled was recessive. So that's the first thing that he figured out, was this principle of dominance. The second thing he figured out, which is really quite genius, he figured out that how on earth, he was trying to think of like a way, a mechanism, how is it that you could end up with a round mama and a wrinkled daddy and then all the babies around? How could that work? Well, he figured out if you had two copies of what he called an inheritance factor. He didn't know about chromosomes, he didn't know about genes. But he said, what if mama has two copies of these inheritance factors and daddy has two copies of the inheritance factors? And then mama and daddy can each give one copy to their offspring, so that this mama is giving an R and this daddy is giving a little r. Well, that might seem really basic to you. You probably figured out that all these babies look like this in terms of their genes. But remember, he didn't know about genes. So what he found was what he called he found or discovered or defined was what he called the principle of segregation. And in this sense, we're using the word segregation to just mean separation. In other words, this parent had two copies, they separated, and she donated one. This parent had two copies, they separated, and he donated one, okay? Now, a couple other things I want to mention is that this round is a physical appearance. Wrinkled is a physical appearance. Round is a physical appearance. In genetics, we refer to the physical appearance as the phenotype. I'm just going to repeat that, pheno. But we happen to know that this round has two of the same alleles. This wrinkled has two of the same alleles. We're going to call anyone that has two copies of the same allele homozygous. That's one word, I just had to make room. And this individual has one copy of one allele and one copy of the other, two different. So this individual is heterozygous. And this isn't its phenotype, right? This is not a phenotype. This is actually what its genetic makeup is. So we're going to call this the genotype. And this is all going to be important later when we move on to learning about some other things later in the lab. Okay, so we know about genotype, we know about phenotype. Now, let's just pretend that we take two of these individuals that have two different copies of the allele. So we're going to have a heterozygous individual, and we're going to mate them with another heterozygous individual. Well, how can we figure out what the offspring look like? You guys probably remember this from your general high school biology days. I find that students usually kind of like these problems. These are fun puzzles. So you're going to draw this square. This square has a name. It's called a Punnett square. It is named for the geneticist. I think his name was Reginald Punnett in 1901, who figured this out. Now, how are you going to label this? Well, what you're going to do is you're going to figure out what gametes this individual could form from meiosis. And if we were to take and split this apart like we would in meiosis 1, you would find that some of the gametes are big R and some of the gametes are little r. Another way of thinking of it is this parent can either give you this copy or it can give you this copy. And we would do the same for this parent, right? And you can fill this in, right? You all remember this. Now, we have a couple things we can deduce from this. We can figure out, first of all, that we have a genotype ratio, just looking at their genes, of 1 to 2 to 1. You have 1 that's homozygous dominant, 2 that are heterozygous, and 1 that's homozygous recessive. That is for the genotype. I'm just going to put G here. That's a genotypic ratio. Let's talk about the phenotypic ratio. So what do these individuals look like? Well, this is a round seed, so I'm just going to draw a perfect circle. This is a round seed, right? Because remember, we got a dominant dude right there. This one is round, and this guy is wrinkled. So our genotypes, there's 2 different ways these guys can look. Our genotypic ratio is 3 to 1, okay? Now, when we are looking at a single trait like this, there's a name for this type of genetics problem. It is called a monohybrid cross. So if looking at a single trait is called a monohybrid cross, you can probably guess what looking at 2 traits at once is. And before we can talk about looking at 2 traits at once for a dihybrid cross, we need to talk about the third principle of a dihybrid cross. And I'll put 2 traits. You guys figured that out. But before we can really understand dihybrid crosses, we need to understand the third thing that Mendel discovered, and that is the principle of independent assortment. And all of this is in your lab. I'm just explaining it for you. So I'm not going to write everything out because it's hard for me to. Let me change my screen really quickly for you. I want to show you your lab, and I want to show you a picture. Oh, golly, here we go. I want to show you a picture from that lab that really explains independent assortment quite well. Okay, so here we are in your lab sheet. And what we have here is, oh, let's see here. I was showing this to someone earlier. Okay, so we have a cell, and let's look at the cell. In this scenario, I've given you 2 chromosomes, not just 1. And notice that on these 2 chromosomes, we have some different genes. So for example, on this chromosome, here we have a gene, big A, little a. Let's just say that this gene is for eye color, okay? I'm making stuff up, okay? And we have big B, little b. Let's say that that is hair color. And then you have another chromosome that has the gene D on it, and let's say that that's skin color. Okay, so we have these 2 are dominant. Let's just say that that's brown eyes, brown hair, and the recessive would be blue eyes and blonde hair. And then here for skin color, let's just have the D stand for dark. So if you have the big allele, the dominant allele, you're dark skin. And if you have the little allele, you're light skinned, okay? Now, if we did meiosis on these guys and we just split them down the middle, then what you would find is one possible gamete would be brown eyes, brown hair, but we have a recessive here, and light skin. And over here, this side would be blue eyes, blonde hair, and dark skin. Now, here's what the principle of independent assortment says. It says, well, we've got these different chromosomes. They may not always line up the same way. What do I mean by that? Let me erase this so you can see. Well, look here. Here you have, I'm going to call this purple, a purple chromosome on the left with a pink on the right, and a pink on the left with a purple on the right. But during meiosis, how these chromosomes line up in your cell is random. So in this scenario, look, you've got pink over here on the left and purple on the right, pink on the left and purple on the right. And if we zipped these down the middle, it's a meiosis 1 scenario, This gamete here looks different than this one does, simply because the chromosomes lined up differently. Here we have, like, what did I say this was? Blue eyes, blonde hair, light skin, and brown eyes, brown hair, and dark skin. So these look different because these lined up different. And basically, all that the principle of independent assortment is saying is that these chromosomes line up independently. Sorry, it's so hard to write on here. Line up independently. So what the heck does that have to do with dihybrid cross Punnett squares? Well, let me show you. Let's go back to my PowerPoint. If I can find it. Nope, that's not what I want. That's what I want. Okay, back to the PowerPoint. So in this dihybrid cross, I want to look at two traits. So we were looking before at seed shape, okay? And we had big R, little r, and you guys already know that that means round and that means wrinkled. Let's also look at seed color, and let's say that capital Y stands for yellow. I'm gonna do these, it's confusing. Stands for yellow, okay? And it is dominant to little y, which stands for green, okay? So let's say that we have this heterozygous individual here. This individual has a capital R, a capital Y, little y. So it is a heterozygous genotype. It's heterozygous for both traits. So I'm just gonna put hetero. That's its genotype. What is its phenotype? It is a round seed that is yellow. Okay, and if you don't understand that, you probably need to go back and reread the lab. So let's look at how we might do a cross looking at two traits at once. Now, when you're doing these dihybrid crosses, you're assuming that this trait is on one chromosome and that this trait is on a second chromosome. So if we look at this in terms of the cell, that means you could have a situation where you have a big R. Each of these is a chromosome, little r. You could have big Y, and you could have little y. But I just told you that how these line up is random. So what if we flip-flopped it? You could also have little r on this side, and big R on that side, and big Y, and little y. And you could go through and do a lot of different versions of this. That is, to me, very cumbersome. But knowing what we know about independent assortment, another way we can look at this is we can say, well, you can have this one match up with that one, right? To make a sex cell that would be a big R, big Y sex cell, whether that's an egg or a sperm, because remember, you're only going to give one copy of this gene to your offspring. You could be big R, little y, right? You could be little r, big Y. And you could be little r, little y. That's getting hard to see. Okay, these are all of the possible gametes that you could form if you have an individual that's heterozygous here. So how would that look in a Punnett square? Well, first of all, you have to draw a pretty big Punnett square, right? And then you would put these all across the top to do your dihybrid cross. Okay, and I'm going to just draw one. Oop. It does not like it when I screen share, sorry. And then right across here, right across here, right across here, okay? Now, if we cross this individual with another individual just like it, another pea plant that's heterozygous for both traits, we would fill it in here like this. Okay, and then you could fill this in like this, R, R, Y, Y, etc. Let's try one down here. Let's try, let's do this one. Big R, little r, big Y, little y, okay? So you can see that this is how you would do a dihybrid Punnett square. Now let's go back to your lab page and I'll show you one that's already filled in. And the reason this is going to be important is you're going to need it for the next exercise, okay? So here is a dihybrid Punnett square, okay? You can see that there are 16 total spaces on this Punnett square, right? So if you count up the squares, there's 1, 2, 3, 4, and you can go on. There are 16 total squares, right? Out of these 16, and these 16 squares actually correspond to different genotypes. Out of these 16 squares, let's do a phenotypic ratio. Let's figure out what all the different types of physical appearances that you could have are. Let's see here. This one is round and yellow. So it's got the dominant form here and the dominant form here. Let's see how many of these are round and yellow. 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 are round and yellow, which is dominant and dominant. Another way you can look in this scenario is you can be dominant with a round shape, but recessive with a green interior color. How many of those are there? 1, 2, 3. Okay. You could also be, let's see here, wrinkled, which is a recessive trait, with a yellow interior, which is a dominant trait. 1, 2, 3. Or there's one individual that's recessive for both traits. They are both wrinkled and green. So the genotypic ratio for this dihybrid cross between two heterozygotes is 9:3 to 3:1, okay? Now that's gonna be important later on when we're figuring out the next thing. So the next thing that we're gonna figure out is, well, first of all, before we go to this, let me just say there are going to be some Punnett square questions at the end of this sheet. And if you are struggling with Punnett squares or anything that I just talked about, please view these videos that I have linked for you here in the lab. All right. Now, the last thing you're going to be asked to do is to use these Punnett squares as a way to make predictions for what genetic crosses will look like. What these Punnett squares are actually doing is giving us probabilities. And here's what I mean. Let's take the scenario here where you have some flowers and the big W stands for white and the little w stands for purple. So white and purple, okay? And if you cross these guys, what you find is that you have half of them that are heterozygous for the white trait and half of them that are purple. So if we had 100 flowers, you would expect, you could predict that 50 of them were going to be white and 50 of them were going to be purple. So if you went through and you counted 100 flowers and you found 50-50, you would know that this flower was following all the genetic rules that I just taught you. But let's say that you went into a field and you counted this flower and you counted 100 of them and you found that 45 of them were white and only 55 of them were purple. Is that close enough to 50-50 for us to still think that this flower is following the rules? What if 40 of them were white and 60 were purple? Is that close enough to say that we're following the rules? Well, it turns out that in science, we do not like to make subjective calls. We want to know numerically, is that close enough to the rules? And what we're really saying is what we found the same, like mathematically speaking, as what we expected to find? The way we can figure that out is by performing a statistical test called the chi-square test. And here's how the chi-square test works. To describe chi-square, I'm going to talk about M&Ms because right now I am just eating like a crazy person. All right, so let's say that in a bag of M&Ms, we have 6 possible colors. You've got blue, orange, green, red, yellow, and brown. Yummy. Now, our hypothesis is that in this bag, there should be equal amounts of all of these colors. So how many M&Ms did we have in this bag? Well, we had, let's see, 212, 147, 103, 50, but if you added all this up, you would find that there were 600 M&Ms. That's a very generous bag of M&Ms. And if there were equal amounts of all of these, you would do 600 divided by 6, wouldn't you? Because there's 6 different colors. And that would give you your expected number that you would see if our hypothesis that these are all equal is true. All right. Well, now let's look at what we really got. We got numbers very different from what we expected to happen. Interesting. So we have a hypothesis that our hypothesis is actually, this is going to be called the statistical null hypothesis. All that means is that we think there's no difference. Our hypothesis is there's no difference between our expected data and our observed data, that any differences we see are just chance error. But there's an alternate thing that could be true, which is that the manufacturer does not put the colors in equal proportion, that maybe they actually give you more blue and orange. So let's test that mathematically. The first step to testing it is to, first of all, figure out what your expected values are, which we've done, okay? The next step in this test is to follow this formula, which is the chi-square formula. All that chi-square means, chi is the letter X in Greek, means it's chi, and squared is just the statistical test that you're doing, okay? So what you're gonna do is for each category, you're gonna take what you observed minus what you expected. So here you've got 212 minus 100. You're going to square that, and then you're going to divide it by the expected value. So you'll take 212 minus 100, so that will be 112, right? You'll square that number, and then once you get that, you will divide that whole thing by 100, okay? You'll do that for each individual category, and you'll add up all of those numbers. I want you guys to try that. If you do it all correctly, you should end up with this number right here. This number right here is called your chi-square value. Now we're going to use this chi-square value to determine whether or not what we see here is actually close enough to our expected value. Like, yeah, what we're seeing is pretty much what we expect, and any deviation is just chance, right? Now, in science, we have what we call a critical level. We say that things are truly different, and it's not just chance if their probability based on statistics of being different is 5% or less, okay? So this is what we call the critical value. In other words, if there's a 95% chance that things are happening due to, let's see, I'm saying that wrong. We want things to be 5% or less due to chance. Let's just leave it at that, okay? So in order to know if this number tells us whether or not these are the same or different, we need one more thing. We need to calculate the degrees of freedom. The degrees of freedom is simply the number of categories you're looking at. 1, 2, 3, 4, 5, 6, minus 1. If you ever take a statistics class, they will explain to you the concept behind degrees of freedom a little bit more. But for now, just know it is your categories minus 1. So our degrees of freedom for this problem are 5. So what we can do is we look at this table and we say, okay, we've got 5 degrees of freedom right there, okay? And we have a chi-square value of 235.42. Well, let's see what we can find on here. Here's a chi-square of 4, one of 9, 11, 13, 15, 20. It looks like 235 is off the charts in this direction, doesn't it? That means if you were to extrapolate this out, this number is going to get smaller and smaller and smaller. The probability that our numbers are the same and any errors are due to chance is really, 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 really low. So what that means is that our assumption that these are put in equal amounts is not true, okay? These are not the same. So here's the way you can operate. If your number that you end up getting as a result of your chi-square test, you can say that they are the same, that what you're observed and your expected are the same. And we're going to use this for a genetics problem. So we're going to say that means that what you saw is following the genetic rules. oops, rules, sorry, can't, there we go. If the number, the chi-number you get for chi-square is bigger than this critical value, okay, then we're gonna say that they are different. And that means that the system you're looking at is not following the rules. Something else is going on. If you ever take genetics with me in the future, you'll find out there's lots of different things that can mess up this system or work differently than what I just taught you. And so you can use chi-square to figure out if the system is not following the rules. So let's apply that to Punnett squares, okay? Now, the reason I showed you that dihybrid cross earlier is it's gonna come into play right now. Let me get rid of this. All right, so this is, let's pretend that we are Gregor Mendel and we went out into our garden and we looked at our pea plants and we expected what we said was a 9 to 3 to 3 to 1 genotypic ratio. If our peas are behaving according to what we expect, we should find a ratio of 9 to 3 to 3 to 1, okay? So let's say we went outside and we observed, we counted up all the pea plants we could find and we found this many were round and yellow, this many were round and green, wrinkled and yellow, wrinkled and green, and we counted a total of 556. Well, that's not what we expected. We expected 313. Well, how did I get that number? Well, 556 is how many we counted, and there were 16 total possibilities here. So let's do 556 divided by 16, right? Let me show you an alternate way that might be easier for you guys. No, I want to do it that way. Okay, 556 divided by 16, right? Because we expected 9 out of 16 to be round and yellow, 3 out of 16 to be round and green, 3 out of 16 to be wrinkled and yellow, and 1 out of 16 to be wrinkled and green. So let's do 556 divided by 16. Let me pull that up for you on my calculator. That number turns out to be 34.75. And then what I would do is I would multiply this number by 9 to get 313, multiply this number by 3 to get 104, multiply this number by 3 to get 104, and then multiply it by 1 to get 35. They've just rounded up here. Okay? Then you do your observed minus expected, you square that, you add that up, this is our chi-square value. All right, 0.510. All right, now our degrees of freedom in this problem is the number of possibilities minus 1. So there's 1, 2, 3, 4 different categories, 4 different ways you can look. So our degrees of freedom is 4 minus 1, which is 3. So let's look this up on our table. Let's see, where's that table up here? Okay, so let me erase all of this. Okay, so I said that it was, our degrees of freedom were 5, right? Okay, yeah, that's right. I said our degrees of freedom were, 3, and our number was 0.5. Where would that fall on this chart? Well, this chart is little on this end and big on this end. We are off the charts in this direction, which means that our data that we observed is the same, mathematically speaking, as what we expected. If that number had been off the charts in this direction, it would be different than what we expected, okay? So what I want you to do is I want you guys to try and perform chi-square using these corn ears here. And the directions are on here. What you're basically doing is there's 2 possible ways that corn can look. Okay, it can be purple, which in this case is showing up as black, and purple is dominant, or it can be yellow, which is showing up here as kind of this gray, and it is recessive, okay? So what we have here is 2 heterozygous parents, okay, that made up these corn ears. And what you would expect is this ratio here. Our genotypic ratio should be our phenotypic ratio should be 3 to 1. We should have 1 yellow for every 3 purple. So what you're gonna do is you're gonna count up these kernels and you're gonna fill in the table and you're gonna perform a chi-square to see if this situation is following what we expect or not, okay? So hopefully this will help you understand the lab. Again, let me know if you have any questions and I will talk to you soon.